Hello, so this is looking at investigating antimicrobial properties of plants. And the two plants we're going to look at are mint and garlic. So, how do we do this? Well, first of all, before we do anything to do with bacteria, to make sure things are a fair test, we need to do something um, called aseptic techniques. So, this is aseptic techniques here. I'm going to put the laser pointer on. Aseptic techniques make sure that there are no traces of bacteria around that can contaminate your experiment. Because remember, you want to make sure everything is controlled apart from your independent variable and what you're measuring, which is your dependent variable. So, how do we achieve aseptic techniques? Well, there's lots of ways to do this, and we need to make sure we use all of them to have the maximum impact to make sure everything is nice and safe and secure. So number one, we need to use a Bunsen burner, or we can use a spirit burner. Basically, we need a flame. So the flame, what that will do is it will increase air pressure around the area and make sure that no bacteria in the air would float into our petri dish, which we'll use later. The next thing we'll need is to clean the desks. So we need to use some at least 70% alcohol solution. We need to wipe our desks or lab benches down with that. Also, the equipment we're using, which we'll come on to later, should also be sterilized with the alcohol. And the last thing we need are obviously we need to protect ourselves and also the bacteria on our fingers. We need to make sure that doesn't contaminate the sample. So we need to use gloves and goggles. And obviously, in a more professional environment, they'll use special suits as well, especially if it's a very dangerous bacteria that they're handling. Okay, so we're looking at bacterial growth. So we probably know that we need to use a petri dish. So this is the technique on how to test if if the plant substance has antimicrobial properties. So basically, can it stop bacterial growth? Here we go. So you're going to have a pestle and mortar, and what you're going to do is you're going to put some garlic in there. So you're going to get one clove, and you're going to pop it into the pestle and mortar. Then what you're going to do is you're going to add some ethanol as well, and you're going to crush it around a bit more until you get a nice smooth paste. Okay. Then what you need to do is get some paper discs. You're going to hold them with the tweezers, and then you're going to dip it into the garlic. Then you're going to let it rest on the side for a bit so that the ethanol evaporates, because we don't want the ethanol to really be a part of the investigation. We're not measuring how much ethanol um, is antimicrobial. We're looking at how much the power of garlic as an antimicrobial. So let the ethanol evaporate. So maybe leave it on the side for maybe a minute. 30 seconds might do fine as well. Don't wave it around in the air because you might catch bacteria on it. So make sure you just keep it still. Then you're going to place it into your petri dish. So you might place it over here somewhere. Then you're going to do the same but with mint. So instead of garlic, you're going to do mint and then place it here. Then we're going to get one more paper disc, dip it in some ethanol, let it rest on the side for a bit somewhere, and then you're going to place it at another point of the petri dish. So you'll have 3 paper discs. Now this petri dish will already be loaded with bacteria. So this is now the petri dish. So the red part is kind of where the bacteria has been spread, and the technician has done this for us already. So and we've put the 3 discs on. So we've got the garlic disc, the mint disc, which is yellow but it looks brown right now, and the control disc. And the red kind of shaded part is where all the bacteria has been spread onto the petri dish. We wait 24 hours and incubate it in our incubator. Then what we do is we have a look at the zone of inhibition. So this is basically the area which bacteria has kind of been killed. What you would notice is that one substance might have a larger zone of inhibition than another one. So you're thinking, okay, how do you measure this? Well, one way to measure this is to measure the radius, and then you can do pi r squared to measure the area of the zone of inhibition, but that does include the paper disc. So maybe we should find the radius of the paper disc as well and subtract that from the zone of inhibition. Either way, what we're trying to do is find a comparison. Yeah. So we're trying to find out which one, garlic or mint, is best at preventing bacterial growth. Obviously in this one, well, I think you can tell, it's garlic. The control has no antimicrobial properties because it's just a piece of paper. But we need a control to make a comparison because these are both pieces of paper. They were both dipped in ethanol, and the only difference between them two is that this has garlic antimicrobial stuff in it, and this one doesn't. So that's why it's good to have that comparison. Okay, and then what you're going to do is probably measure it with a ruler, and then that's it. That's all you need to do. In terms of disposable, we give these back to the technicians, and then they'll get rid of them in the correct manner. Thank you very much.